This is Dr. Ben Weitz with the Rational Wellness Podcast, bringing you the cutting-edge information on health and nutrition from the latest scientific research and by interviewing the top experts in the field. Please subscribe to the Rational Wellness Podcast on iTunes and YouTube, and sign up for my free ebook on my website by going to drweitz.com. Let's get started on your road to better health. Hello, Rational Wellness Podcasters. Thank you so much for joining me again today. This is Dr. Ben Weitz. For those of you who enjoy listening to the Rational Wellness Podcast, please go to iTunes and give us a ratings and review. And for those of you who'd like to see Dr. Robillard's face as well as listen to him, you can always go to our YouTube page and watch the video version of the Rational Wellness Podcast. Anyway, our topic for today is irritable bowel disorder or syndrome and small intestinal bacterial overgrowth, and then how to treat it. Irritable bowel syndrome is a condition marked by gas in the intestines, bloating, abdominal discomfort, constipation, diarrhea, alternating one and the other, as well as a series of other symptoms. IBS, irritable bowel syndrome, is the most common gastrointestinal disorder and occurs in up to 20% of the U.S. population. For many years, IBS was considered a stress-related condition with no known cause. And this is partially because IBS was most common in women. Traditional medical treatment has been generally composed of medications for controlling symptoms, such as a medication to reduce constipation, another medication to control diarrhea, et cetera, et cetera. But in functional medicine, we don't want to just treat symptoms. We're trying to get to the underlying cause of our health conditions. Dr. Mark Pimentel from Cedars-Sinai was really the first one to discover that small intestinal bacterial overgrowth, or Sibo, is the cause of IBS in up to 84% of cases. Sibo consists of bacteria that normally grow in the large intestine or colon that then start to grow into the small intestine, which normally has relatively small amounts of bacteria. When you eat certain types of carbohydrate foods that contain fermentable fiber, our gut bacteria eat the carbs and they produce various types of gases like hydrogen and methane. When this occurs in the colon, this is not a problem since the colon is very expandable and there's a valve, the ileocecal valve, to keep this gas from going back up into the rest of the digestive tract. But the small intestine is a relatively narrow tube and is not really expandable in the way that the large intestine is. And if there are bacteria there that are eating fermentable carbs, they'll produce gas, and this gas likely will cause discomfort, a feeling of gas and bloating, and many of the other symptoms of IBS or Sibo. The gold standard for diagnosing Sibo is finding more than a tiny amount of bacteria upon jejunal aspirate, which means that when putting a scope down into the intestines, you scoop a small amount of the liquid in the proximal jejunum, which is the lower part of the intestine, and then analyze it via culture or PCR. Unfortunately, this test is very invasive and is not typically done on a regular basis in clinical practice. So what we have is a somewhat imperfect but fairly useful test, which is known as the lactulose hydrogen methane breath test to diagnose Sibo. Dr. Pimentel also found that Sibo often results from a bout of food poisoning, where the bacteria that causes food poisoning give off an endotoxin referred to as cytolethal distending toxin. The immune system reacts to this toxin and attacks this cytolethal distending toxin. But then, because the cytolethal distending toxin is similar to some of the structural proteins in the intestinal wall, there's the immune system cross-reacts and ends up attacking the intestinal wall. And this is by this understanding, Sibo is really an autoimmune condition, and this can be diagnosed via a blood test that Dr. Pimentel developed called the IBS check test, and now Cyrex Labs has Array 22. That's a more sophisticated way of trying to measure these antibodies produced by the immune system in this attack on the on these toxins, in order to be, as well as the antibodies to the structural proteins in the intestinal wall, in order to be able to diagnose Sibo. This is all obviously a very complicated and confusing condition, and that's why I've asked Dr. Norm Robillard to join us today. Dr. Robillard's an expert at IBS and Sibo. He has a PhD in microbiology from the University of Massachusetts, where he studied bacillus species of bacteria, which are the spore-based bacteria. He's the founder of the Digestive Health Institute, and he's also a gut health expert, author, and microbiologist. He's the creator of the Fast Track Diet and the Fermentation Potential System, the author of the Fast Track Digestion book series, and the publisher of the Fast Track Diet mobile app. Thank you for joining us, Dr. Robillard.
Thanks for having me on, Ben.
So, when you look at the studies on IBS, there's a big range as to what percentage of patients with IBS are actually positive for Sibo. And so there's one review paper that says it ranges from as little as 4% all the way up to 78%. And this has led some practitioners and doctors and researchers to even wonder if there really is a relationship with Sibo and IBS and what percentage it is. On the other hand, Dr. Pimentel did a study in which he found that 84% of patients with IBS tested positive for Sibo. Why do you think there's such a wide variation in these results? And what percentage of patients with IBS do you think are really caused by Sibo?
Yeah, that's a good question. Of course, there's basic test methodologies. I used to run an analytics testing laboratory. And so there'll always be precision, accuracy, intermediate precision, which is how closely, different laboratories get the same result. So it requires a lot of training and a lot of calibration to get these, labs on the same page. And of course, the test for Sibo, there are different techniques used. Some people, you mentioned lactulose sugar, which is really, I would think, the preferred method because lactulose is not digested by humans, so it passes through the intestines until it runs into bacteria that can break it down and produce hydrogen. So when you use lactulose, you can detect bacteria all the way through the small intestine. But a lot of labs use glucose, which is quickly absorbed. And so if the bacteria are not up in the proximal or early part of the small intestine, you may not even see them with that test. So it's less sensitive. However, it's more specific. So there are some test methodology issues. Before we went live, you and I were just talking about the test preparation, which I think is also important and potentially kind of a flaw or an imperfection in this testing, because before people take the breath test, there's a 24-hour preparation period where you're essentially on an intervention diet. And so you're avoiding legumes. You can still have some fish and chicken, but they don't want you to have too much, no fruit juice. And you mentioned, they can have some rice, and some rices are worse than others, as we'll get to in a little bit. But nevertheless, they're on an intervention diet for 24 hours. And then they fast overnight before taking the test. And if, you obviously follow Dr. Pimentel's work, and those folks down there have really worked a lot on this migrating motor complex. So Sibo is a snapshot in time. If these bacteria work their way up into the small intestine, your body, your stomach acid, a lot of these mechanisms we'll be talking about today, motility, it's always trying to move these bacteria out. Bile, antimicrobial factors, your immune system. And so the more chance you give it to not be either fed more with carbs, things like fasting, the more of a chance you have of being negative. On the other hand, if you feed everybody a lot of fermentable material, you're going to see the number of people with Sibo go way up. And again, it's a snapshot in time. And then the last point I wanted to make is, and I hope we get a chance to talk about this a little bit today. I had coined the term LIBO in an article I wrote a few years ago on resistant starch. To stand for large intestinal bacterial overgrowth. And in your introduction, you mentioned, well, as long as all this fermentation happens in the large bowel, you can accommodate that all bacterial growth and a lot of gas. But if you have too much, that could be problematic as well. So people could come in with symptoms and test negative for Sibo, and they still have excess bacterial fermentation. That's what I believe. So the second part of your number was what's the number, right? What should it be? 87% or 4%? And, again, from being in analytical testing all of these years, I do know that if you test something enough, the answer gets closer and closer to the accurate value. And so, and Dr. Pimentel had put together a meta-analysis of a whole variety of studies and really validated the finding of Sibo and connecting it to IBS. And I thought that was pretty powerful. So when you look at enough data, you're going to get a better answer. So is it 45% or 65%? I don't know, but it's probably somewhere in there. And again, it fluctuates.
Right. Yeah, that's interesting. You talk about LIBO or bacterial overgrowth in the large intestine. I wonder if you could use the Sibo breath test to diagnose that if you just look at the rise in gases after, at 120 minutes or beyond.
Probably not, because when you go from even somebody with Sibo, they have greater than 100,000 bacteria per mil in the small intestine, which anybody that works with bacteria will tell you that's such a tiny, tiny amount. Right. And then you get to the large bowel, and the large intestine contains 100 trillion bacteria, so much more. So basically this lactulose would be just rapidly consumed and produce a lot of gas, even if somebody didn't have, quote, LIBO.
Oh, okay.
However, there was a study done, kind of jumping into this LIBO thing a little bit, by a group that looked at, I think they were out of Johns Hopkins, looked at 47 patients with IBS and some controls. So it's pretty good end for statistical analysis. And they had them swallow a wireless motility capsule, kind of a smart pill. And this pill could detect pH differences. So it was going through the stomach, small intestine, and into the large intestine, and it was measuring acidity. And what they found was really interesting. They didn't see any pH change, no increased acidity in the small intestine. And just to back up for a minute, when bacteria ferment carbohydrates, they produce short-chain fatty acids, right? They're fats and they're acids, and that's where the acidity comes from. But they didn't see any increased acidity in the small bowel of people with IBS. But when the smart pill got to the large bowel, they did see an increase in acidity. And they indicated that they interpreted that as the bacteria in the large bowel for IBS patients were very active, producing more of these short-chain fatty acids and more of the acid. So they were questioning Sibo. And while we're on that topic of questioning Sibo, there was another lab up in Canada that used a radioactive tracer molecule when they gave people, and again, it was a pretty good sized study over, I think it was about 40 people with IBS and some controls. When they gave them the lactulose breath test, at the same time they gave them the lactulose, they gave them this radioactive tracer probe. And they could literally follow this probe on somebody's body, and they had kind of mapped out, here's where the end of the small intestine, the ileocecal valve should be right here. And they were literally following this radioactive probe through the small intestine. And what they found was that by the time people were registering as hydrogen positive within 90 minutes, indicative of Sibo, that dye had already reached the ileocecal valve and was entering the large bowel. And so again, they were saying that they think that people that test positive with Sibo perhaps have faster motility. And that's what they're measuring, not the actual Sibo. So I thought about both of these studies, and I really think the answer is that both LIBO and Sibo exist. For instance, in this radioactive probe study, yes, as people were recording a positive breath test, some of this dye, for at least 5%, had reached the large bowel, but a lot of the dye was still back in the large intestine, small intestine, and a lot of the lactulose was still back in the small intestine. So I don't think it debunks Sibo so much as it suggests both of these studies that we need to look at LIBO and Sibo. And then while we're on that topic again, with the SMART pill and the acidity, they found that they had no change in acidity in the small intestine, but they did in the large bowel. But again, getting back to the numbers, even if you have 100,000 or 500,000 bacteria in the small intestine, is that enough bacteria to meaningfully change the pH, especially when the small bowel adds bicarbonate and it neutralizes the acidity from the stomach and all of that? So I think Sibo could exist, easily exist, and that it's not debunked by these methods, but I do think it is a good reason for us to look at it in a different way and think maybe it's a combination.
Yeah, and a thought I also have about that, and then I think maybe I should ask you to explain what the Sibo breath test is for those listeners who are not familiar with it, is some of the real acid-loving bacteria like the lactobacillus are typically not part of the Sibo equation, whereas primitive archaea are, and I wonder if they're not really as acid-producing as some of the other bacteria, so that could be a factor as well.
Well, in Sibo, so we've been talking about breath testing, but and you also mentioned the gold standard, which is taking an endoscope that can sample the small intestine aseptically as possible, pull these bacteria out and try to culture them. And some of those studies have been done. And they're not perfect studies because, first of all, 80 or 90% of the bacteria in your gut will not grow in culture. They haven't figured out how to grow them. And that's why they use the 16S RNA gene sequencing to look at these strains molecularly. But they have done some culture work, and they find that Sibo is comprised of an overgrowth of some bacteria that are from the small intestine. So there's your lactobacillus and some staph and strep, but also bacteria that are more associated with the large bowel. Some Firmicutes, some Clostridia species, some Bacteroidetes like Bacteroides fragilis, and several other gram-negative and gram-positive strains. So bacteria from the large bowel and from the small bowel are overgrowing there. So that
and also the archaea, right?
So that's some people may have thought that early on because in some of these breath tests, the people have high levels of methane, and they're like, wow, okay, must be the archaea. They're not bacteria. These other microbes called archaea take the hydrogen and they use it to reduce carbon dioxide to methane. That's the little molecular food chain there. And so where are these archaea? Because wow, even in the first sample, they have high methane, and they just continually have high methane. But remember, with these breath tests, they have you blowing in these tubes, right? So that if any hydrogen is being produced by bacteria in your gut, gets absorbed into your bloodstream, exhaled through your breath, and you capture it in this tube. You blow in that first tube, it's called time zero. Put the cap on, put a label on it. That's time zero. That's before you drink the sugar solution. Then you set that aside, and then you drink the lactulose sugar solution. And then every 15 minutes, 20 minutes, depends on the test, usually about every 15 minutes, you blow in a new tube and put the cap on. And with hydrogen, you can see a real kind of time course there if you plot it out. Zero sample, there's hardly any hydrogen, and then all of a sudden it starts to come up, and maybe 40 minutes and 60 minutes comes up, then it starts to maybe go down a little bit, and then it hits the large bowel past 90 or 120 minutes, and it goes through the ceiling. So you can get a profile like that with hydrogen, but with methane, you almost always just see that it's high in the 0 time point. So that's telling us that it has no dependence on the lactulose sugar. It's doing its thing, taking the hydrogen and CO2 and combining that to make methane. And there has been some work on it. I'm not sure I can cite any particular studies at the moment, but that shows that these archaea are in the large bowel and that they're just kind of churning away. So if you have, and some people may not know this, people with IBS-C, constipation predominant IBS, almost always have these really high levels of methane. And because methane, there's been good work on this. You can inject methane into the intestines of animals and it slows down the transit. And there's just such a tight correlation with people that are high methane having slow transit and constipation. So, but there is a strong belief that these archaea organisms are doing that in the large bowel.
But I thought that they had grown into the small intestine, and that was one of the theories about, I talked about Dr. Pimentel's theory about the autoimmune component, but to follow up on that, what that meant is by damaging the structural proteins in the small intestine, it caused decreased motility, and he describes it as like a stream that stops running quickly and starts backing up, and that allows the bacteria from the large intestine to grow into the small intestine. So I thought that's where the archaea.
Well, right. So, this whole story, and it's a great story, by the way, they worked out with cytolethal descending toxin and the autoimmune reaction with vinculin, right. Slows down motility, hits the nerves, and so forth, causes constipation. But slowing everything down, whether that causes archaea organisms to back up all the way into the small intestine, I'm not sure. But I do have my own theory about connections between LIBO and Sibo.
Okay.
Something we're not talking about today, but it's just relevant in this discussion, is when I first got into nutrition and dieting, it was only because I had chronic acid reflux myself.
Right.
And I had found that a super low carbohydrate diet caused my symptoms just go away. And so I was so amazed by this, and I was playing around with this idea, and I started following the food groups through the digestive process and came up with this new theory that what was happening, I believed, was that I was consuming too many carbohydrates, too many were getting malabsorbed, feeding blooms of gas-producing bacteria, right? As a microbiologist who grew these bacteria, right? You mentioned I worked in a bacillus lab for my graduate work, but I also worked as a postdoctoral fellow on Bacteroides fragilis. That's 10% of the gut bacteria, and E. coli. And I was actually the first one to be able to move genes between E. coli and this strict anaerobe Bacteroides through this conjugative process. But one thing I knew about these bacteria, they produced a lot of gas. They were saccharolytic. They loved carbohydrates. They produced a lot of gas. And I came up with a theory that all this gas produced, too many carbs, was pressurizing my stomach. It was translating into my stomach. And they do know people with GERD, acid reflux, have much higher pressure in the stomach. And the theory was it was pushing reflux, opening this valve. Instead of the original theory that stood for 60 years was saying that this valve was dysfunctional, or it was weaker, or it was relaxing spontaneous. And so there's a lot more evidence for this gas producing by bacteria driving reflux. And so I've written a couple of books on that. Now, to this new discussion on Sibo versus LIBO. These guys with the pH Smart pill, they found all this acidity past the ileocecal valve in the early part of the large bowel. And how could that relate? Suppose you had Sibo and LIBO. How's the Sibo getting there? And I have a theory that it might work the same way as my acid reflux theory, that these bacteria are producing a lot of gas in the early part, the ascending colon, just past the ileocecal valve, a lot of growth, a lot of gas, and maybe this same gas pressure is pushing back on the ileocecal valve. And it's interesting, it ties into another study done out of Johns Hopkins as well, I think, where they found that people with Sibo, was it a Sibo population or an IBS population? I think it was Sibo, but one or the other, but probably people with IBS, with Sibo. They found that their ileocecal valve pressure, and they measured it by, with like a colonoscopy tube going right up to the valve, and they put a pressure sensitive, like a manometry instrument, position it right in the ileocecal valve. And it was weaker. There was less pressure, people that had Sibo. And so it could be the same thing. If you have gas pressure from bacteria pushing back on that valve and pushing it open, if that you have a pressure sensitive tube in there, it's going to look like it's weaker, but really it's being forced open by back pressure. So anyway, something to think about.
That's interesting. So, you know, if the archaea are really in the large intestine, that could be one of the reasons why it's difficult to correct methane Sibo and kill or you know, cut back the archaea if rifaximin, which is what gastroenterologists often use when they treat this, is basically acts in the small intestine. So it might not be acting in the large intestine where, under your theory, the archaea really are.
Yeah, well, it probably explains why rifaximin alone is not efficacious for IBS-C.
Right, that's why they usually recommend rifaximin plus neomycin or another antibiotic.
Yes, and both are non-absorbable. If you've seen my other work, you know I'm not a big fan of antibiotics, but at least these two are non-absorbable. You can eliminate some of the systemic problems.
Right.
But they use both. The rifaximin is probably, by their own estimations, not very useful in the large bowel, right? There's a whole story about, well, it won't upset your microbiome because it requires bile for its most efficient inhibition. And 95% or more of the bile is reabsorbed at the end of the small intestine. So the rifaximin is probably not a big factor. So it would be really, what's the neomycin doing?
Interesting.
Yeah.
So we've talked about a couple of the things that could cause Sibo. We've talked about the damage to the motility of the intestinal tract, and you mentioned the migrating motor complex. And you also talked about the ileocecal valve. What are some of the other causes of Sibo?
Yeah, motility is big, and Pimentel thinks it's really probably one of the biggest. Low acid is another one. People with hypochlorhydria, achlorhydria. You know, the acid, not only is it important for digestion, but it's important for keeping the bad bacteria out of your gut, keeping the bacteria in your gut from moving up to your throat and lungs and sinuses. So acid's important. And by the way, on the acid, that you see that in people that have, there is an autoimmune disease that will lead to atrophic gastritis and hypochlorhydria, but it's quite rare actually. So unless somebody has a lot of other autoimmune conditions, you might not have to look at that one. But definitely PPIs, that's what they do. They knock down your stomach acid. And the other big one is a prolonged infection with Helicobacter pylori. It's bacteria that infects the stomach. It's a corkscrew shape like a spirochete, and it burrows down through the mucus and anchors on the stomach lining, and it makes these colonies. And depending on where those colonies are in your stomach, that's where the damage happens, and that's where the gastritis and atrophic gastritis happens. So some people, it affects the hormones that regulate stomach acid, and they can have too much stomach acid, and they're very susceptible to duodenal and stomach ulcers. People that have these bacterial colonies of H. pylori near the parietal cells that produce the acid, those are the ones you have to worry about having low stomach acid. That's a big one too, I think, for in a subset of people.
That's really interesting, because most people think of H. pylori as automatically associated with increased acid production, but you're saying if the H. pylori grows in a certain part of the stomach, it can be associated with decreased hydrochloric acid secretion.
Absolutely. Right. And the two doctors down in Australia, one of them gave himself an H. pylori infection and got gastritis.
Marshall, right.
Oh, that was a great story. But they were focused on the ulcers, you know, with the cause of ulcers. So but they can also cause the opposite. And the people that have low stomach acid, not only will it really mess with your digestion, you can still have symptoms, but they also are at higher risk for stomach cancer with the low stomach acid, which caused me to wonder if they studied PPIs, if they would see there was also a gastric cancer risk with PPIs. None's been reported so far, but also really knocks down your stomach acid. So just to cover a couple of more quick ones, any kind of liver issues. We talked about liver, and it produces the bile, and that's antimicrobial. People with cirrhosis, any kind of liver problems, they can have Sibo, a lot of problems. Anything with your pancreas, pancreatitis, right? The pancreas produces amylase, protease, and amylase, protease, and lipase, right? So if you have any kind of problem with your pancreas, you won't be digesting food as well, especially if you're deficient in the amylase. Even kids with cystic fibrosis, while they don't have a pancreas problem per se, they do have a lot of mucus in the ducts where the enzymes are released from. So kids with CF have a very high incidence of Sibo and GERD for that matter, right? There's another link there. And they have to be on digestive enzymes. Problems with drugs, we talked about PPIs, pain medicines. Man, there's a whole story about NSAIDs. It's just unbelievable. Too many pain meds, especially narcotics on the motility front. Pimentel had, in one of his interviews, was talking about anybody on morphine has Sibo. You don't even have to ask. So these things, they really do slow down the motility. We talked about GI infections already, and not just bacterial. Gastroenteritis from food or waterborne illnesses. Bacteria, yes, but also protozoa, viruses, anything that causes gastroenteritis.
Even fungal infections, and then you can have what's called a SIFO, or a small intestinal fungal overgrowth.
Certainly, sure. I can't remember if we talked about some other. We talked about the ileocecal. Other genetic based diseases, celiac and Crohn's, it's huge. Diabetes, it might be a nerve damage issue going on. Surgery and adhesions, I've heard Pimentel talk about that. I cover all of these in the Fast Track Digestion books, by the way. I have a whole chapter on this. Speaking of scarring, scleroderma is a big problem for a lot of people with Sibo. But here's one that never gets any attention, and I'm not sure why. Too many fermentable carbohydrates in your diet. I learned that the hard way myself when I was in my early 40s. I was having a terrible time eating all these carbs and having all these symptoms. Took the carbs out of the equation, and no problem. So I really think that some people, maybe as we get a little bit older, a lot of these functional GI issues start in our mid-30s and 40s. Our digestion just may not work quite as well. You might not be able to put your finger on exactly what it is, but if you're not digesting and processing carbohydrates well, digesting these things is a real finely tuned collaboration between our own digestive prowess and the ability of these bacteria to help us out. And if we overwhelm them and throw in a couple of these other potential underlying causes, I mean, you're in real trouble. So there's a handful. I have a whole chapter on this if people want to read more.
Yeah, it's interesting. I think a lot of us in the functional medicine world, when we are putting patients on treatment protocols, I know myself, we usually use a diet that's designed to have less fermentable carbohydrates. I typically use the low FODMAP diet. Now, you've looked at some of these diets like the low FODMAP diet, the specific carbohydrate diet, some of these other diets that are popular, and you found some problems with those diets, and so you came up with your own system. Maybe you could tell us about your Fast Track diet and your fermentation potential figure for being able to analyze quantitatively which foods to include.
Sure. And the story goes back quite a few years. About 15 years ago is when I really found that very low carbohydrate helped my GERD symptoms. Started looking into this. And so that's not on your list, right? You mentioned FODMAP and specific carb diet, but just a low carb, even a ketogenic diet. I use ketogenic diet in my own consultation practice as a kind of a troubleshooting tool. And we'll get into the Fast Track diet in a minute, but it allows some carbohydrates, kind of a flexible approach for people with different dietary preferences. Jasmine rice is better than basmati rice, for instance, for reasons we can talk about. But what happens if, even the jasmine rice, I'm having IBS symptoms or heartburn symptoms? Okay, well, maybe until there may be a problem for you even digesting the easier to digest starch, for instance, jasmine rice has amylopectin, an easier to break down starch. Some people have trouble even with that. So I'll go to a ketogenic diet just as a troubleshooting method to say, well, let's take all the starches out and see how you do. But so why did I come up with this diet? Well, initially, I had just found a low-carb diet works, but when I came up with this theory about the underlying cause of reflux, linking it to bacterial overgrowth, similar to what Fermentable was doing at the same time with IBS, I was doing with acid reflux. But when you limit all of the carbs, it seemed to be okay. So I had written a book on just this mechanism, and I just wanted to get it out there. It's a new theory, new way to look at acid reflux. And I was living not too far away from you at the time in Thousand Oaks, California. And I was pretty close to Dr. Mike Eads, who was living down in Santa Barbara. And his book, he and Mary Dan, his wife, also an MD, who wrote Protein Power, their book was the first time I ever read about any kind of diet ever. And it was to go on this low-carb diet. And I noticed in his book, which he had written in the mid-90s, he said, oh, by the way, we have patients that their heartburn improves on a low-carb diet. And I'm like, that guy speaks my language. So I sent him my book on the mechanism. And well, actually, I didn't just send it to him. I was going to send it to him, but when I had him on the phone, I said, gee, I'm going to be at the farmer's market down in Santa Barbara. We agreed to meet. And I gave him a copy. And so we became friends, had a little, drink some wine together, and we were talking about this stuff. And he was the one that really asked a key question. He said, well, low carb is helping heartburn. We know that, and now we know there's a mechanism for it. And he bought in fully into my theory. He thought it made sense, and there's a lot of evidence for it. But he said, which types of carbohydrates are the most problematic? And that one question sent me on my way for another couple of years of research. And I wasn't really aware much of FODMAP and the specific carb diet. I think I had at some point read Elaine Gottschall's book, Breaking the Vicious Cycle. I can't remember exactly when. So I was aware of that. But I was just thinking about which of these carbs are hard to digest. So I didn't have any specific, lenses on except just looking at these. So I came up with lactose, fructose, and of course, polymers of fructose. Those are actually dietary fibers. Resistant starch, fiber, and sugar alcohols. So there's 5. And the next step was to come up with a way to quantitatively measure these in foods, because how do you pick up a pear? How much of these 5 things are in that pear? Who knows? You can give it to the best dietitian in the world. She might not know either, or he. So I was thinking about this problem. And by the way, the specific carb diet, it limits disaccharides, it limits grains and starches, but not honey. So that didn't cover these 5 that I was interested in. And the same with the FODMAP diet, which, fermentable, oligodimonosaccharides, and polyols. So these, some of the monosaccharides like fructose, polymers of fructose, sugar alcohols, polymers of galactose for that matter too, but not fiber or resistant starch. And I think the reason not all these diets cover it is because they either just don't think some of these are very fermentable, or they just think they're so darn healthy for our gut that we need to include them. But I came up with the 5. I came up with the FP calculation to quantitatively measure them. But many years after I wrote the first Fast Track Digestion book, I came across this textbook. Can you see that?
Yeah.
Textbook of Primary and Acute Care Medicine. It's fat.
Wow.
A lot of good stuff in there. But, this book was published in 2004. It's used to train doctors. But I think some doctors might have skipped the chapter on intestinal gases, because when you go to page 1192 and open it up and read it, there's those 5 carbohydrates that the Fast Track diet restricts. So it's not that far out there. It's aligned with this textbook of Primary and Acute Care Medicine. So that's why I chose those 5, and why I developed my own diet that was low-carb keto is a great approach. The other ones, I think they're missing a couple, and I think that can be challenging for some people if they're having problems with there's so many different types of fibers. And we know some of those are very fermentable, stachyose, raffinose, various other polymers of sugars. So I think you need to limit all 5. And then the other thing was I needed to find a way to do it quantitatively so I could make the diet something people could use. And so in the books, in the mobile app, the Fast Track Diet mobile app, there's all these tables of all these foods with these FP values. And the FP calculation, it took me a long time to figure that out because I didn't know how I was going to measure these 5 carbohydrates in a way that you could just look at any food and say, is there a lot of fermentable material in this or not? And I struggled with that issue. And for a while, I started thinking about the glycemic index. And started thinking, well, that measures how quickly carbohydrates go into the bloodstream. Must be some way to use that. And I was just, I don't know, kind of dense thinking about it at first. But as I thought about it more and more, eventually I realized that it wouldn't be that hard to modify that equation, kind of flip it around and modify it, because instead of measuring carbohydrates going into the bloodstream, I wanted to measure how many carbohydrates were persisting in the small intestine and not being absorbed. So I turned the equation around, but because the glycemic index equation does not measure fibers or sugar alcohols, I needed to add those back after. So I flipped the equation around, and then after that first part of the calculation, you add dietary fiber and any added sugar alcohols. And so all you need is the glycemic index and the nutritional facts for any food, and you can do this calculation. And of course, the app has a calculator for this, but also it has tons of table sets. We're releasing a new version in the next month. We're about 10 months behind releasing this, but it's going to have over 1,000 foods in it now. And it's got, voice recognition on it. So if you just open it up, I have it opened up now, search, whoops, I pushed on the happy face instead of the other button. Carrots. You can bring up carrots raw, and you can cook them after, by the way, but it will just tell you what the fermentation points are for any given serving size. And if the points are too high and you still like carrots, just use a few less. If you use half the number of carrots, it will cut the servings, will cut the points in half. So that's how it works in a nutshell.
Is there a quantitative amount of FP points somebody's supposed to have in the course of a day, or is that related to their total caloric intake, or how does that work?
No, that's a good question. It does matter a great deal. In fact, we're listening to our readers on this one. We had initially set, a flexible range between 25 and 35 points. And just to put that into perspective, 30 grams of undigested carbohydrates that are fermented by bacteria can allow bacteria to produce 10 liters of gas. So just an ounce of these carbs can drive a whole bunch of gas production. That's why you do need to limit these. Typical Western diet may have 150 or more FP points a day. So that's 50 liters of gas. It's a lot. And so we want to get that level down. And so we recommend somewhere between 25 and 45, depending on if you have a lot of severe symptoms, you want to go to the lower end. And as you get better and start improving, you can increase up to more than 45, but you probably never really be eating 150 a day again if you're somebody that suffers with these functional issues. Now, we have a Facebook group, Fast Track Diet official Facebook group. People should join. There's about 8,500 members on there right now, and sharing recipes and talking about all this stuff. And we have a lot of groups of people with these different conditions, not only IBS and Sibo, acid reflux. We have people with laryngopharyngeal reflux, real subtle irritation in the throat and vocal cords. It's linked to reflux, but it's subtle, but it's also persistent. And it's hard to get rid of. And so we've had people on the page, many of them saying, well, this irritation's persistent, subtle, but you know what? If I really was diligent with my points, I had to go less than 20, down to 15. And I had to do it for weeks, and some people, months. And finally, their throat symptoms would get better when nothing else worked. But they were driving this with us, and they were saying, you know what? You're not cutting the points enough. We have to cut more. So we're learning from them, and we're involved in a clinical study where we've actually reduced the points based on what people on our Facebook group are telling us.
Interesting. I wonder if there's a issue with pre-diabetic or diabetic patients, because your program is actually promoting consumption of foods that have a higher glycemic index, right?
Well, I never thought about this as promoting. It is a flexible eating plan, right? And so, yes, if you were going to have rice, you rather than Uncle Ben's or basmati white rice, because a small bowl of those is going to give you, a whole lot of points, maybe 10, 20 points, depending on how big the serving size.
And that's partially because those foods are digested slowly, right?
Yes.
And it makes them more fermentable.
I'm making your point for you. Yes. Whereas the jasmine or sushi rice has a higher glycemic index, hence a lower FP. They're more easily digested. More will go into your bloodstream, less will stay behind. So you'll have less GI symptoms. Now, what happens though, your blood sugar increases. And so in the book, we're cautious to warn people about prediabetes, metabolic disorders, and diabetes. And people have to be responsible for their own blood sugar levels. Somebody that doesn't have an issue, or they're an athlete, or they're carb loading, or they're a construction worker. So this book, a lot of different people are going to use this book. Other people may have to really watch their carbohydrates. So low GI carbs is one way to do it, but it is going to feed the bacteria a lot. So that's the downside. And if you've got a functional GI issues, your solution in the end will probably be a lower carbohydrate diet. That way you'll have less blood sugar. You'll also feed less of the bad guys in your guts, the overgrowing bacteria. So we're not telling people to load up on sugars, even if you look at our tables and the serving sizes. There's different categories on this app. So if let's just go down to rices, right? And so, well, here's the one we just talked about. I'm going to click on jasmine rice, cooked jasmine rice, all right? What's the total serving size? I don't know if you can see it there, but it's a half a cup. People with blood sugar issues, chances are they're wolfing down a lot more rice than that. So we purposely, even though it's low in FP points, we purposely tell people that really small serving sizes are better, lower carb is better, and eat slowly and chew well, which will also help digest these starches and some of these carbs better.
I was surprised to see wine and beer or light beer on your list, because these are fermented, but I guess there's a difference between foods that are fermented and are fermentable. Is that the case, or?
Okay, so yeah, you're bringing up a couple of points there. Foods that are fermented, right? There's less carbohydrate in those foods when you consume them, because the fermentation happens in a vessel, in a vat or a tank or a mason jar, right? And these lactic acid bacteria in the case of pickles, the yeast in the case of beer and wine, they're using the sugars and they're producing alcohol, right? Well, not in the lacto-fermented, they're producing short-chain fatty acids and so forth, but in the beer and wine, they're producing alcohol. And they're consuming the carbohydrates. So when you consume those foods, there's less carbohydrates than there otherwise would be in there. However, when it comes to beer, a light beer has many fewer points. I think it's somewhere around 6 or something for a bottle of light beer, 4 to 6 maybe. No, maybe 3 or 4. But when you have a heavier beer, like one of my favorites, IPA, it has a lot more points because there are a lot more carbohydrates in there. So regardless of the fermentation process, you still have to look at, anyway, it's going to take me too long to find the drinks and open up the beers, but
yeah, I got it.
Yeah. But I purposely, watch my points on days when I'm going out with my buddies, because I want to have a couple of IPAs, but they have more points. And so if I'm also eating French fries and something like that, and I have a couple of these beers, it's kind of like death from a thousand cuts. These points add up, and then they really get you. And then you need a couple of days, 2 or 3 days, to unwind it. So you have to pick your poison. But it is great that that distilled liquors are fine because they have no carbohydrates at all. Dry wines, red or white, are pretty low in points, and light beer is pretty low in points. So from there, you just have to, conserve your points and pick your poison.
Now, it's common in the functional medicine community to put a patient on one of these restricted diets, along with using some other protocols to try to get rid of the Sibo. And yet, once we're done with the treatment period, whether that be 1 month, 2 months, 6 months, whatever that period is, it's usually recommended that we try to broaden the person's diet as much as possible. And this is to make sure that we are bringing back in some of the fibers that are necessary to have a healthy microbiome, and also to make sure we're getting all the phytonutrients from having a diverse diet. But you have a little bit different take on using your fast track diet, don't you?
Well, I do. I've been doing this for 15 years myself, and my own, which was terrible, chronic acid reflux. I mean, it was horrible. I was choking in the middle of the night, reflux entering my lungs, and all day heartburn. It was a terrible situation. So I've been doing this for 15 years. I don't worry so much about encouraging people to add back fermentable material. I find that when people get better, and the more you do stay low FP or control these fermentable carbs, or focus on identifying and addressing all of these underlying conditions. Like a lot of people with H. pylori, that will say, well, I'm just not going to do anything about it. I heard it's not that big of a deal. Maybe when you're younger, but in time, gastritis, loss of stomach acid, you have to, and you might need some help. We have a consultation program where we really work on this. What are these things, and what are the risk factors, and what are your symptoms? How do we mostly throw things out, but the things that are remaining, the few things that are remaining, we have to confirm and address those. And of course, I'm a microbiologist, I'm not a doctor, so I give them my notes. I'm a consultant microbiologist to them. They take my notes to the doctor, and we work through these things. You have to identify and address potential underlying causes. That's one thing. But as you reduce the fermentable carbs, work on the underlying causes, you're going to have less fermentation, less bacteria there that make proteases that damage the enzymes in the tips of your villi, toxins. You may actually damage the microvilli and villi themselves, kind of like a mini version of celiac disease where you get some blunting. It does occur with Sibo. So as you have less inflammation, control your diet, look at these causes, your digestion will improve. I do like to encourage people I work with to broaden their diet in terms of 3 food groups. Low FP vegetables, and there's a lot of those. If you look at the vegetable lists in the apps, there's, 180 vegetables, fresh herbs. But low FP fresh herbs, low FP fresh vegetables, because with the idea of a diverse diet, they do have some fiber and some fermentable material, just not as much. But diversifying it will diversify your gut microbiota. So fresh vegetables, fresh herbs, and then also some small servings of some fermented foods, lacto-fermented pickles, kimchi, sauerkraut, that kind of thing, maybe a little bit of yogurt. You just don't go overboard, but some. So that's a nice mix. But this thing about immediately feeling like you have to come back with a ton of fiber, I think that's misguided. I think this, I might be one of the only ones out there at this point arguing against too much fiber. Everybody just seems to think we're starving our microbiota. I just don't believe it, especially if you have an animal-based diet with some fatty fish and you still have plenty of green leafy vegetables and you're consuming some nuts. That's, to me, that is an ancestral diet and a healthy one. So I feel like people, when they get better, they can be the best judge of what they can tolerate. I feel like I don't have to ram fiber down their throat. And, on that topic, I was reading a review by, his name's William Che. I haven't met him yet, a gastroenterologist up in Michigan, who I hope to meet at this upcoming Sibo conference, because he's doing a GI guy doing work on diet. It's just great to see. But he wrote a review, co-authored it with, I think, one of the Australian folks that works in these diets, on fiber. And they are on the same page with me. They recognize there's so many different types of fiber, and the fermentability is so different. And so they work their way, well, what's the fermentability of all of these diets? I have a chapter on that in the Fast Track Digestion books where I do the same thing. And they reach the same conclusion. Psyllium, cellulose, something like that's less fermentable. And if you're going to play around with anything, maybe start there. That's the less invasive of all of these other ones, because some of the other ones are very fermentable. And there's papers on using these with people that had GERD. They gave them fructose oligosaccharide, right, a polymer of fructose, but it's a mini dietary fiber. And their reflux, they were measuring it with probes, and the reflux occurred much more frequently. It was much more severe, and they had terrible symptoms. So that's a prebiotic. They gave people with GERD a prebiotic, and they really almost killed them. I mean, it was terrible symptoms and terrible reflux. So,
and that's very common in the functional medicine world right now. Is prebiotic supplements, and you even see doctors saying, rather than use probiotics, much more important to use prebiotics, and it's definitely very popular right now.
Yeah, it is, and I'm not against prebiotics in very small amounts or a little bit of experimentation. Some people are worried about taking a probiotic, because some of these probiotics contain a prebiotic, with the idea that will help get these bacteria going once you swallow them and they get into your intestines. But if you look at the label closely on those, they typically add about 50 milligrams of one of these prebiotics, which is really a tiny amount. It's less than 1/10 of an FP point, because an FP point is a gram. 1 FP is 1 gram of fermentable material.
Yeah, I think part of this, putting the prebiotics in afterwards, also after your treatment period, is sort of part of the 4R or 5R program that's been so prominent in the functional medicine world, and pretty much accepted as, one of the few, biblical verses in the functional medicine world, which is that first you get rid of the bad bacteria, and then you replace and repopulate with bacteria and prebiotics, with probiotics and prebiotics.
Yeah. I mean, my favorite way to repopulate is just to have an organic garden in a compost pile. That's what I've been doing my entire adult life. Flipping the compost. I'm growing my own garden, so I get away from the chemicals for a lot of the year.
Yeah.
And we're harvesting some squash right now. We'll put them in the basement. They'll still be good next spring. If you make some pickles, those will last you into the winter. So there are ways to eat less, chemicals. And then also, when you eat some of your vegetables raw, maybe not all of them. Cooked vegetables are a little bit easier to digest, but at least some of them raw. Some greens. We grow a lot of dill and parsley and basil. Then you are repopulating your gut with bacteria from your environment. And by the way, they've done studies on compost piles. There's a lot of similarities between the microbes in our gut and what's in a compost pile. A lot of the same, groupings of bacteria. So that would be my preference. Yeah, I'm not a real nut for the prebiotics, and unless they're in limited amounts.
Right. I'd like to ask you one more question, because I know we need to wrap up soon. From reading some of your articles, you're generally not recommending a lot of nutritional supplements. You do recommend digestive enzymes and ox bile and probiotics, but you don't particularly like herbal antimicrobials. And I think these are a common part of many functional medicine protocols for Sibo. I know myself, we typically put the patients on one of these restricted diets and include these antimicrobials. And the thought is, first we're going to kind of starve the bacteria, and then we're going to try to kill them using natural agents like berberine and oregano oil, et cetera, et cetera. And but you're not a big fan of these.
Yeah, I mean, I can see why there's a temptation to do that. I'm not totally against it. I spent 10 years working on and developing antibiotics. I worked on the development of Cipro. I've studied mechanism of action of antibiotics. I've studied mechanisms of resistance and the genetics of antibiotics. So I've worked on them for a long time. They can be lifesavers. I mean, they're very important. I'm against the loose use, I guess, of antibiotics because of resistance and a lot of problems. I really want to see diets continue to be improved and refined. And a lot of people have eating disorders or preferences, and it's hard for them to change the diet. I understand that. I'm a foodie, you know, I hear that a lot. I want to eat what I want to eat. I get it. But, you know, I have more of an ancestral health perspective. And I've been reading more and more about this over the years, and it just makes so much sense that the more we can eat like our ancient ancestors, that's really the way we evolved. But of course, it's not exactly the same. Today, the foods aren't the same. The Western diet is just not only terrible, but also the availability of all of these snack foods. It's just too easy to eat these things. And so it's hard to change a diet. But also, our microbiota is changing, especially since the invention of antibiotics and the clinical development of antibiotics in the 40s. That's only been 60 years, and it's having a huge impact on our gut. Also, chemicals and preservatives. So our gut bacteria are not the same. They're not nearly as diverse. If you go back and look, and Jeff Leach has done some great work living with and eating like and sampling the guts of the Hadza in South Africa. Their guts are much more diverse. They're much more in touch with the biosphere than we are. And so we've got this gut microbiome that's gotten used to eating more processed foods, more easy to digest food. And then all of a sudden we're getting all this advice. Well, if you want to be like the Hadza, you better throw some of this fiber at it. But your gut microbiota is not, and your digestive tract is not handling it well. And so you can keep fighting it, or you can just say, well, I have to go with it a little bit, but I still want to eat healthy. Green leafy veggies, occasionally maybe a half of a sweet potato or a half a cup of rice for some people. You also have to consider, you mentioned diabetes, but also how about cardiovascular risk? It's huge. I don't know if you follow the work of Ivor Cummins and Jeffrey Gerber. They just came out with a new book that's super, and follow the lectures.
What's the name of the book?
Eat Rich, Live Long, I believe. Yeah, I just started reading it myself, but yeah, you can also Google Ivor Cummins' lectures, just phenomenal stuff.
Okay.
Really looking at things, starting out from the basics and saying, what's important? Is LDL really that important? Turns out, actually, it correlates very poorly with cardiovascular risk. But you know what correlates really well? Insulin. So it's just fascinating lectures. I can't say enough for their work.
But I'm not sure if you're distinguishing between antibiotics, which are prescription medicines, and oregano oil. You know, I totally agree that antibiotics, especially broad spectrum antibiotics, have a negative effect on the microbiota, and can have harmful effects. But generally speaking, my understanding and my reading of the research is that these herbal antimicrobials don't have a negative effect on the microbiota.
Yeah. Okay. So I didn't cover that. Let's cover that. First of all, when you look at the history of antibiotics, most of them came from other living things back in the day anyway, from bacteria, from fungi. And then, the Germans started to figure out how to make, used chemistry and came up with sulfur drugs and so forth. And then it, grew from there. So a lot of regular commercial, antibiotics come from other organisms. So do these herbal antibiotics. And while I would say you could put them maybe on a less invasive scale than some of these, more powerful, pharmaceutical antibiotics. Every antibiotic is going to kill or inhibit, right? Some are cytal, some are bacteriostatic, are going to kill or inhibit a certain variety of bacterial types. And they're also going to have a certain potency, right? What is the concentration of antibiotics you need to get to kill those particular microorganisms? So with herbals, and there is that one study, was that also, I think that was Johns Hopkins as well. It's funny, all these studies from Johns Hopkins, talking about using, berberine and some other herbals, and that they were as good as rifaximin. So I think that's interesting, and it's good to look at, and maybe they're not as bad, but we don't really know that much about it. A lot of the work for those has been done kind of on the side, outside of the mainstream. And I do think we need to learn more. And by the way, when you say something as good as rifaximin for treating Sibo, you're basically saying, if you look at the target studies, that's 10% better than placebo. So I think diet, behaviors, and identifying and addressing underlying issues, those three things should be front and center. If you don't do that, I think you're dead in the water. After that, then you can start to look at supplements, digestive enzymes, absolutely. If there could be a stomach acidity issue, betaine, right, some vinegar, work your way down. Somewhere herbals are in there is something to try, but for me at least, and for people that I work with, I just don't know enough about it to really be too gung-ho.
Have you used motility agents like ginger, 5-HTP, things like that?
Yeah, I think again, it's, there's a whole thought that motility is slow because something's wrong, right? And that could be, and yes, with the vinculin and the gastroenteritis, absolutely. But there may also be an adaptation to, depending on what you're consuming, right? Because bacteria want to get all those calories, help us get all of those calories out of the food. Our bodies are collaborating with the bacteria. So depending on what you're eating, your motility could speed up or slow down for a whole variety of reasons. It doesn't really mean something's absolutely wrong. And then also you do have to look at the extremes of diet as well. I was reading a study the other day that in anorexics, they found that they had a spike or an increase in these archaea organisms, Methanobrevibacter smithii, that produces the methane. So it makes me wonder, under extreme caloric deprivation, is this a mechanism to make sure you wring every last calorie out of any kind of vegetative matter you consume by these archaea going up and motility slowing down? So I first try to look at everything in terms of what could the natural mechanisms be here, and am I smarter than 50 million years of evolution? No, So I just have a little more of a cautious approach. I try to really understand before I just jump to try this and try that.
Awesome. Thank you, Dr. Robillard. This has been an amazing podcast. Gives us a lot to think about. How can listeners get a hold of you and get your books and your programs?
Sure. Well, you can find us at digestivehealthinstitute.org. And I would also encourage people to join the Fast Track Diet official Facebook group. I'm on there most days, poking in here and there, answering a few questions, but it's a lot of people that have become real experts in the diet, and they're very helpful as well. So I think those are the two main places to find us. If you want to specifically look at the mobile app, you can go to fasttracked, TRACT, diet.com, but you can find those on iTunes and Android store as well.
That's great.
Thanks for having me, Ben. It's great talking.
Thank you, Norm. Yeah, I really enjoyed the conversation.
Me too.